Hello, everyone, and welcome to today's webinar, Linear DNA, Improving rAAV Production and Synthetic DNA. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. I'd like to invite everyone to participate by submitting your questions during this presentation. To do so, simply type them into the Ask a Question box and click Send. If you have any trouble seeing or hearing this presentation, simply click on the question mark icon on the left toolbar for further help or submit those questions in the Ask a Question box. So let's get started. Today's webinar will be presented by Dr. Jude Samulski, the President and Chief Scientific Officer and co-founder of AskBio. Dr. Samulski received his Ph.D. in medical microbiology and immunology from the University of Florida. His graduate work demonstrated the first use of AAV2 as a viral vector which culminated in the first USA FDA-approved gene therapy drug for Leber Congenital Amaurosis, typically referred to as LCA. Now, Dr. Samulski has worked with AAV for 40 years and has advanced therapeutics in human clinical trials for hemophilia, Duchenne muscular dystrophy, giant axonal neuropathy, Pompe disease, and congestive heart failure. 450 articles and holds more than 300 patents related to AAV technology. For a complete biography of Dr. Jude Samulski, please click on the Biography tab at the top of your screen. Dr. Samulski, welcome. You may now begin your presentation, sir.
Thank you, Susie. Very much appreciate it. It's a pleasure to be here today. I will take about 30 minutes to walk you through an innovation that we think is going to be impactful for the production of recombinant AAV vectors. I'll start out with a little bit of history to level set everybody as to the topic and where we originated from a number of years ago. And I'll bring you into focus with respect to the problems or the concerns that have emerged as this technology has been marching into the clinic. And then hopefully leave you with an expectation of what's likely to come based on the synthetic DNA approach. I want to first mention that Lester Suarez was the proposed speaker today, and he was not available because he was getting this COVID shot. So as a result of that, I'm going to step in and assist with his presentation. So I'll first start with giving you a brief introduction of the history of AAV. AAV was discovered in the 50s in an attempt to identify what was causing the common cold, and they were isolating adenoviruses from the adenoids of patients and were interested in generating a vaccine that could be used to protect individuals. Advance. And as you can see in this magnification, the small particle there is actually an AAV particle, Advance, that was eventually discovered based on the life cycle and utilized as a method to determine that it was an independent virus, Advance. As you can see in this cartoon, it's about a 20 nanometer particle compared to the 100 nanometer adenovirus, and it ended up being a virus that was non-enveloped and carried a single-stranded DNA genome, Advance. Here's a cartoon illustrating the AAV life cycle. On the right-hand side, you see what's referred to as the lytic life cycle. And if you advance, you can see that in the presence of adenovirus, both viruses will infect the cell and generate progeny. However, if AAV infected a cell in the absence of adenovirus, as illustrated on the left-hand side, it would be genetic suicide, but instead the virus devised the method of latency where it could persist in a dormant state. However, if this cell ever was infected by advancing, you would see that it would re-enter the lytic life cycle and produce progeny. So from an AAV vector perspective, if you advance, you see that what we were able to do in the late 70s, early 80s, was to clone AAV into a plasmid backbone. And as a consequence of that, if we took this inert plasmid DNA and introduced it into a human cell and then co-infected with adenovirus, we were able to reproduce all of the steps of the AAV replication cycle. So this was the initiation of the AAV vectors. And if you advance to the next slide, you can see, if you advance one more time, what's basically happening is in the laboratory, one is able to remove 96% of the AAV genes, which are referred to as rep and cap, and replace that with a therapeutic transgene. On the left-hand side of the slide, you can see an AAV capsid cartoon, and so at this point, the AAV vector is packaged into the proteinaceous shell and ends up carrying the new sequences into a target cell type. If you advance, you can see that this was first demonstrated in 1989 in a paper published in Tom Shank's lab at Princeton, where we separated the cis-acting elements illustrated by the dark boxes in the plasmid at the bottom from the trans elements, which are represented by the plasmid at the top. If you transfected these two plasmids into a human cell and once again infected with adenovirus, you would turn on the replication genes and the capsid genes, and they would rescue the cis element and package the foreign DNA into an AAV particle along with adenovirus. And so these were the early progressions of trying to uncouple the AAV life cycle so it could be utilized as a viral vector. If you advance 2 times, you'll see that the AAV transgene at this point could simply be packaged into any AAV serotype. If you advance, and advance, you'll see that one has the ability to put AAV into many different flavors of AAV. And if you go to the next slide, you'll see that when you take these different serotypes of AAV with a so-called reporter gene, such as luciferase, and inject it into the muscle of a mouse, that they transduce at different levels of capability, where you see AAV1 is much better than AAV2. And if you advance your slide, what we essentially were establishing at that point, if you advance, is that many different genes could be substituted into these proteinaceous shells. And if you advance one more time, you can see these were basically building what we refer to as molecular FedEx trucks. If you go one more slide, you'll see that at a little bit later time, in 1998, we were finally able to uncouple the adenovirus dependency and extract out of the 36,000 base pair genome 4,000 nucleotides that were responsible for helping AAV go through its life cycle. So now you had 3 plasmids being introduced into the human cell, and after 48 hours, only one virus came out, which was the AAV vector of choice carrying the therapeutic sequences. So in the next slide, advance one more time, you'll see that what ended up happening was many research groups were beginning to take different AAVs, as seen if you advance AAV1 for muscle. If you advance, you see AAV2, which was approved for Leber Congenital Amaurosis, as Susie explained earlier, and AAV8 were being used to target genes to the liver. All became specific carriers for therapeutic proteins based on the production systems I just described. And if you advance one more time, you'll see on the left-hand side that what has happened is the field has advanced. There's many plasmids that carry various capsid proteins, one adenovirus helper plasmid, and at the bottom, a plethora of AAV therapeutic genes that are clumped between the terminal repeats. These are all introduced into a bioreactor, but in essence, what we really need, if you advance, is on the right-hand side. We only need the rep and cap gene in the adenovirus helper sequences, as well as the vector. So as a consequence, what ends up happening by default is that we end up with a lot of the so-called plasmid backbone as a contaminant. And this contaminant has CPG motifs that are insults to innate immune response. They carry an antibiotic resistance gene, and they potentially carry neoantigens that could be problematic in patients. So why do I focus attention on this? Let's go to the next slide and I'll explain. There are basically three things that seem to impact AAV production at the moment. The first one is illustrated here as time, and if you advance, you'll see that in order to make GMP plasmid for manufacturing, one has to establish what's referred to as a master cell bank, or MCB. It takes about 3 months to do this, and then if you generate so-called FDA GMP plasmid, that's about another 5 to 7 months. If you advance further, you can see that the cost for making this, and if you continue to advance, is hundreds of thousands of dollars per gram of plasmid. And then finally, if you advance and advance again, you'll see that contamination ends up being a byproduct of this current method for generating these core resources that are required for production. So let's go to the next slide and ask a question: why is there contamination in the AAV preparation? And I'm showing you an illustration here of a cartoon that shows the AAV inverted terminal repeats illustrated by these T-shaped hairpin structures flanking either side of a reporter gene. And in the bottom, I'm illustrating on the left-hand side the contamination of plasmid sequences inside of the virion compared to the therapeutic vector on the right-hand side. And you can see we get anywhere between 1 to 10% of the plasmid backbone is being packaged as a contaminant. Now, if you follow the logic behind the cis-acting sequence, or these terminal repeats are responsible for packaging, you have to scratch your head and say, why is the plasmid being packaged at any level? It doesn't appear to or should not have terminal repeat sequences. And if you advance to the next slide, you'll see a paper that was published by my colleague Zhao, who was my first student, and his wife Jean Li, where he was able to show that when you put in an infectious clone of AAV into a human cell in the presence of AD, what actually happens first is what's referred to as a resolution step. This is where you get a recombination across these inverted terminal repeats. And as a result of that, you generate two products: an AAV product, as expected, with the terminal repeats of interest, and a plasmid that has a portion of the AAV terminal repeats attached to it. When these are introduced into the production system, the AAV sequences that are the correct ones amplify. However, the plasmid sequences still carry AAV packaging elements on it, and as a consequence, these inadvertently get packaged into capsids. Zhao was able to show this by generating a plasmid with only one ITR that he referred to as double D, and as it resolved itself into a linear plasmid with ITRs on either side, he was able to show that this could be packaged very efficiently. So we knew the cause. We understood what the mechanism was. The question was, how do we resolve this and get away from this potential contamination? Around this time, our colleagues in France and elsewhere were beginning to show that when you take the AAV vectors and you sequence them using next genome sequencing, they were able to show that there was at least between 1% and 3% of contaminants that were showing up. And if you advance, you'll see that I'm referring to down at the bottom, a DMD patient today at 30 kilograms would potentially get about 3 times 10 to the 14 vector genomes per kilo, which is about 10 to the 15 vector genomes that are carrying bacteria sequences. And this is somewhat troublesome if you advance to the next slide, because you'll see that in an example where AAV2 was used to put factor IX into patients with hemophilia, if you advance, there's about 10% of the input vector was contaminating with plasmid sequences. So right off the bat, we have an issue. Although the system was very flexible and very dynamic and very quick to change different serotypes, it presented itself with a contamination that to today is still being tolerated by both the regulatory community as well as the research community. Let's advance to the next slide and see what our options are. The options to solve this problem were actually done some 35 years ago in a publication in the Journal of Virology, in which we engineered a very specific plasmid that, as you can see in the cartoon in the middle, that at the very bottom of the illustration of the ITR, it was designed to carry a unique restriction site. This unique restriction site was PBU2, which did not cut in the AAV genome. And if you advance, you'll see that what we have on the left-hand side, if you advance again, it's a circle illustrating where the unique restriction sites were. And they surgically removed the plasmid backbone away from the AAV packaging sequences. As illustrated on this, there's a restriction site, claw 1, that's a unique cutter. So if you advance, what you'll see is that we took 3 different substrates, the PBU2 surgical removed plasmid backbone, the linear AAV vector or the circular AAV vector, and we introduced all of those into an AAV replication system. And if you advance one more time, you'll see the results of this where it illustrates very graphically that the AAV genome that was cleaved away from the plasmid sequence actually replicated more efficiently and generated large amounts of viral genomes, suggesting that if one could artificially remove this resolution step, one could generate more material in the end and basically a cleaner material. And if you go to the next slide, you'll also see that slide labeled Back to the Future. We were able to show that in this specific plasmid, there was a phage polymerase origin of replication, and that if you infected these cells with the phage, you would be able to amplify this plasmid backbone, and that if you purified it, in this case it's being purified as a single-stranded genome, and digest it with the PBU2 enzyme, you could again separate the AAV sequences away from the bacterial plasmid. This, however, was complicated by the fact that it was all done in E. coli and it generated single-stranded genomes, and it wasn't until present-day technology that we were able to revisit this concept and actually utilize it as shown in the next slide. On the left-hand side, you see once again the rep and cap plasmid illustrated below. You also see that when you resolve the rep and cap plasmid, about 4,000 nucleotides are represented by the rep and cap gene, and about 3,000 come from the plasmid backbone. If you advance, you'll see that today there has been an innovation in the technology where one can add a telomerase sequence at the end of the AAV ITRs, and as a recognition of the sequence by a phage polymerase, one's able to, again, surgically remove the plasmid backbone from the AAV sequences of interest. And you ask yourself, how do we do this? How it's shown in the next slide. And if you look at the 6 nucleotide recognition sequence, this sequence is recognized by, if you advance, the Phi29 polymerase, and it starts to amplify on a rolling circle replication mechanism, a very high fidelity amplification. And if you go advance again, you'll see that the pro-telomerase will then recognize this cleavage sequence and will cut and seal the ends. As a consequence of that, you now have linear DNA that's closed at either end. And then if you advance, you can see where by a cleaning up technology, one can remove the plasmid sequences by DNA degradation. And as a result, if you go to the next slide, you see there's a very strong product being generated as illustrated on an agarose gel. This product is the AAV molecule with closed ends and completely absent of any of the bacterial sequences. Now, the power of this is that all of it's done in a test tube, and there's no bioreactors or contamination with a plasmid at any level. If you go to the next slide, you'll see a cartoon that illustrates the so-called telomere sequence of 28 bases, followed by the ITR, followed by the transgene of interest. And this molecule is now linear, as I illustrated in the earlier manuscript, similar to the PV2 digested AAV sequences. Here we have a linear molecule that has closed ends, and as a byproduct of that, we have removed all of the plasmid backbones, the so-called CPG islands, and contaminants that one has become accustomed to in today's production. If you advance even further, you'll see that by the same mechanism of Doggybone resolution, on the left-hand side, if you advance, you'll see the original concept of how AAV resolves out of the plasmid, and those plasmids amplify. And if you advance again, there's still the ITRs attached to the plasmid sequence, and that gets packaged based on packaging size. However, on the right-hand side, you see our so-called Doggybone resolved material, and I'm only illustrating this by showing the right-hand end of the genome, in which the ITRs are held together by this 28 nucleotide telomere element. And if you resolve this by advancing, you'll see that there's again resolution, and if you advance again, you'll see one product is the AAV genome of interest, similar to what's on the left-hand side of the slide. And if you advance again, you'll see the other byproduct is a terminal repeat attached to this telomerase element. And as a consequence of that, we have basically removed all of the contamination of the original plasmid backbone by this in vitro replication system. If you go to the next slide, you'll see, compared to the left-hand side, which are the original triple transfection helper plasmids we know of today, we're now asking questions with the so-called Doggybone identical materials, just minus plasmid backbones, and they're being tested in the bioreactor. And if you advance, you can see the virus that comes out of these systems, referred to in this slide as dbDNA versus plasmid DNA, are generating almost equivalent, if not higher amounts of so-called AAV vector. If you advance to the middle, next slide, next image, you'll see a silver stain illustrating the AAV products that are being made by Doggybone compared to plasmid are essentially identical. And if you advance one more time, you'll see by Western blot, it's carrying the AAV structural proteins VP1, 2, and 3, and at the right ratio. So from a core element that's being introduced into the bioreactor, everything is coming out identical with respect to yield, quality, and composition. If you go to the next slide, it's an illustration of next genome sequencing, where you can see the blue color shows the percent of contamination that's in the plasmid prep compared to the red sequence, which shows that the sequences completely stop at the end where these termination elements are, and thereby dramatically removing the potential for contamination, as was illustrated by the cartoons. And if you advance one more time, excuse me, you can see that if you take a capsid such as AAV9 carrying a reporter gene such as luciferase, either made with Doggybone or from plasmid, and put it into a reporter cell line such as HeLa RC32, you can see on the right-hand side of the slide that within the expected period of time, we're getting transduction and expression of the luciferase gene. Once again, these data suggest that the Doggybone material is of a higher quality and potency. If we go to the next slide, this is the same vector now put into an animal model. You're looking at luciferase expression after an AAV8 recombinant vector is injected into the tail vein of mice. Compared Doggybone on the left-hand side to plasmid on the right-hand side. And once again, the two final products are performing as anticipated. Again, at this point, it suggests that Doggybone DNA may generate vectors that are a little more potent. And this is being evaluated as we go on. So if we go to the next slide, I think what you're left with as a take-home message is that the standard plasmids that the field uses carries two AAV ITRs cloned into a bacterial plasmid backbone requested by the FDA to be kanamycin resistant, and that when this is introduced into the production system, one generates the AAV molecules, whether it's a self-complementary AAV as illustrated on the left-hand side, or a traditional single-stranded AAV as illustrated on the right-hand side, plus the plasmid backbone sequence as a contamination. And if you advance your slide, you'll see that the standard Doggybone DNA is essentially a linear molecule that has two ITRs on either side of the sequence of interest, and the 26 nucleotides required for the polymerase to resolve and close this in an in vitro reaction. Once again, through the resolution, you can generate a traditional single-stranded AAV as illustrated on the left-hand side of 4.7 kb or a cell-complementary molecule. And at the same time, if you advance once more, you'll see that what we have achieved here is basically reducing the cost, the time, and the product quality of AAV using a synthetic DNA starting material. And so I'll close by showing one last slide. The conclusions and take-home message is that if you analyze these reagents for plasmid backbone, you can see that when you compare the original approach to the Doggybone, there still is the ongoing concern of contamination of plasmid backbones. However, this is not seen with the Doggybone since there's only 56 nucleotides total on either side of the starting reagent. The plasmid production system currently used requires fermentation and growing of bacteria. On the Doggybone, we're only using an enzymatic like PCR reaction in a test tube. The time required with respect to generating this takes months to do the traditional fermentation and purification, whereas the Doggybone is done within days of 2 weeks. We also see that the AAV titers are equivalent, if not superior to the plasmid approach, and that transduction, at least at these early stages, seems to be generating a much more potent reagent. Further studies are being done to evaluate this enhanced transduction, whether or not the contamination of plasmid DNA interferes with vectors, suggesting a lower transduction efficiency. And when you look for the de novo genome, there's none in the plasmid than there is in the Doggybone. So what we're hoping to say in this tutorial is that a new technology has advanced out of the community, supporting research that was done years ago, but allowing one to synthesize DNA in the test tube to generate all of the starting material that's required to produce AAV vectors, which are seeing a lot of success in the clinic, as well as new approaches for treating many diseases from neurological to muscle to bleeding disorders. And I'll go to my last slide and stop and acknowledge that AskBio is a wholly owned subsidiary of Bayer, which has a production component called Virogen that's in San Sebastian, Spain, that generates all the AAV vectors that are used in our clinical trials. It's also a manufacturing facility for anyone who wants to generate AAV vectors to go into the clinic. The Doggybone technology originated from Touchlight. Touchlight is a company in England that developed the polymerase manufacturing in E. coli, and we thank our colleagues at GeneWiz who we collaborate on a regular basis. Thank you, and I'll stop there. Take any questions.
And thank you, Dr. Samulski, for that great presentation. We will now start the Q&A portion of the webinar. And if you have any questions you want to ask, please do so now. Just put them into the Ask a Question box and click Send. So let's get started. It looks like, Dr. Samulski, we already have some great questions coming in. Are there any challenges you have encountered when using RCA amplification compared to bacterial fermentation? And the second part of that question is, how robust is that amplification process?
Very good question, and deserves more attention. Typically, when we do the procedure today under the conditions that have been optimized, we can generate about 1 gram of the so-called Doggybone material in about 3 days' time. That is for either the helper plasmid, the adenovirus plasmid, or the vector. As a consequence of that, the footprint is very small, so about the size of an office is all that's required to generate these GMP material. And it's anticipated that this will only scale to a higher level as we get more proficient in managing the enzymes in the reaction. So it's a very good question, and at this point, the early days of the so-called bumpy road problems have been ironed out mostly so that it's at a large-scale production capability.
Thank you so much. Our next question, one of the challenges faced during AAV manufacturing is low vector concentration. Does dbDNA approach affect the concentration of the rAAV produced?
Yeah, so we are seeing about a 2 to 3-fold increase in the vector that's coming out of the Doggybone production. It appears that by removing the plasmid backbone, it allows the production of AAV to initiate much quicker, more efficiently, and every genome is participating in replication. So in the end, we ended up, it looks like we generate more virus. We've been successful in keeping these viruses at very high concentration after purification by our team at AskBio and Virogen. And so at the moment, it appears that there is a bump in an increase in production, and it also appears that, as was illustrated, these are identical to the material generated with plasmids, but seems to jumpstart the whole AAV production stepwise because you remove the resolution step that seemed to be critical.
Thank you so much. And again, I want to thank our audience for these great questions coming in. Dr. Samulski, what do you see as the next quantum leap in the gene therapy space?
So as you can imagine, there's a significant effort put into production to, one, drive down the cost. Plasmids today make up at least 1/3 of the total cost, and we think that the Doggybone approach of the synthetic DNA is going to significantly decrease that. The turnaround time will also be increased since it takes only a week or so to generate gram amounts of your production material. At the same time, there's a significant effort being focused on generating what they refer to as next generation AAV capsids that are more efficient at going to a target tissue or being able to introduce the gene more efficiently into the disease organ. However, one of the things that we're focusing on, and I think it's an important parameter, is the ability to do what's referred to as repeat administration. Being able to give someone a therapeutic as a young adult or an infant, and as they grow older, if they do need therapeutic levels to increase based on size, one could do repeat administration. Currently, your immune system sees the AAV virus the same as your body would see the COVID vaccine, and it mounts an immune response. We and others are trying to develop technology that will allow these vectors to be given multiple times. So I think that's going to be a significant transformation to the clinical community when that's accomplished.
Thank you so much. And we have time for one more question. In the luciferase expression studies, why do you think the DB-DNA constructs yielded significantly higher expression levels than plasmids?
That's a good question, and it's a curiosity that we're still struggling with. We think there may be less of the so-called incomplete genomes or partial genomes that are packaged by AAV, and therefore you may have more intact and only intact genomes. With the resolution from the plasmid, sometimes you get shearing or smaller DNA genomes that are eventually packaged, and they may just be titered as AAV particles carrying the full length, but from an infectious perspective, they're not complete, and therefore the overall potency of the titer is lower. And so our typical method of using quantitation by genome copies may be giving us a false impression of what the actual transduction titer is. And we're still exploring that rigorously to see if that's what's happening at these early stages of characterizing Doggybone virus compared to the traditional plasmid virus.
Dr. Samulski, I want to thank you again for your presentation, your time today, and your important research. And before we go, I also want to thank the audience members for joining us today and for their interesting questions. And I want to let you know that the webcast will now be available on demand through April of 2022. We hope you've enjoyed this event. Take care, everyone, and have a great day. Bye-bye.
And I would like everyone to know that Lester owes me $5 and a beer for doing his presentation today. Thank you, guys.
Thank you again, Dr. Samulski. Bye-bye, everyone.
Good night.